Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, Engineering the Future of Cancer Immunotherapy, Breakthroughs in CAR T-Cell Technology. I am pleased to welcome Dr. Saba Ghassemi. Dr. Ghassemi is an assistant professor in the Department of Pathology and Laboratory Medicine and serves as a principal investigator at the Center for Cellular Immunotherapies at the University of Pennsylvania. Her research focuses on developing novel CAR T cells with enhanced potency through a multidisciplinary approach that integrates engineering with CAR T cell immunology. A warm welcome to you, Dr. Ghassemi. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And without further ado, I will now hand over to Dr. Ghassemi to begin her presentation.
Thank you so much for the introduction. I will going to start and I will update on advances that we have on generating better CAR T cells and more potent CAR T cells. So this slideshow is the overall process that we generate CAR T cells for adaptive immunotherapy, a process that T cells get isolated from the patient and they get cultured over time ex vivo. Typically, we use like 9 to 15 days in a very nutrient-rich environment. And they're genetically modified with a chimeric antigen receptor. And the highlighted area in red is the area that we're very interested in. And the overall goal of that we have is to increase the potency of CAR T cells. And we have very good reason that this area is very suboptimal, the way that we ex vivo expand the T cells. The success of CAR therapy depends on several parameters that they intersect with each other. For example, if you use CAR design with different co-stimulator domain, it impacts the persistence of CAR T cells, or there are factors in the tumor microenvironment that impact the success of immunotherapy. But again, as I mentioned in the previous slide, our goal is improving the quality of the product, the CAR T cells that we infuse to the patient. And when we talk about quality, generally we mean their differentiation status or metabolic fitness, that these also intersect with each other. For example, if you have CAR T cells with better differentiated state, they generally have better metabolic properties. The current manufacturing process relies on activation of the T cells via their TCR receptor. We previously showed that activation through TCR drives a progressive differentiation over time in culture, which means that after several days, here I put an example from day 0, which is like before doing anything, from the apheresis, and then 9 days after, all the cells are phenotypically and functionally inferior compared to the early days after 9 days expansion. As seen in the graph here, you could see that by day 9 we have complete loss of some of our very important subsets, such as naive-like T cells that are important determinant of the efficacy. And in general, when we get the cells, whether they're like patient cells or healthy donors, they are composed of several subsets, but towards the end of their expansion time, they are more effector differentiated progeny that they lack quality. So they are good killers, they clear the tumor, but they lack quality and they don't persist, which is that means that they're like less effective for T-cell based therapies. And for example, in our NAM6 mouse model of leukemia, after we established the xenograft, we tested this idea. We infused the cells that are expanded for shorter time. For example, here we tested the cells that are expanded for either 3 days or 5 days or 9 days, and tested their anti-leukemic activities in this mouse model. Here we use a subcurative dose so that we use a stress test to see the impact of the quality of the CAR T cells. And here I have to mention that, this last graph, the day 9 cells are our FDA-approved Kymriah process. So this is our standard process, and this is compared to cells that are expanded either for 3 days or 5 days. And we can see here that cells that are expanded for 3 days are the only cells that they can control the leukemia, and based on this BLI message of the BLI measure of the tumor. So and day 5 so have some intermediate response, and then day 9, like they completely lose the control of the leukemia. And this data is confirmed by also the number of the T cells in the blood. You could see that, the number of the T cells in cells that are expanded for 3 days, they're like a lot higher number of T cells compared to cells that are expanded for 5 days or 9 days. So these data, based on this data, we can conclude that if we shorten the ex vivo culture of CAR T cells, then this process would yield like cellular product that are less differentiated T cells, so they have significantly enhanced effector function. And founded on these insights, a clinical trial at Penn tested the efficacy of 3-day product that instead of standard 9 days manufacturing product, we only expand them for 3 days. And we also use the humanized CAR-19 that co-express IL-18 as well. And we tested this in a clinical trial, and there are 2 important factors that I would like to mention about this clinical trial. One, that patients that got admitted to this trial, they must have had prior CAR therapy, meaning that they must have tried one of these either commercialized CAR therapy or within a clinical trial, and they must have been relapsed or refractory to this prior CAR therapy. So technically, this patient, they must have had CAR before, and it must have not worked for them. And this is the interim result, as you can see, and now the complete result is out because the trial is ended, and we had such an impressive response in this clinical trial. And by now, I think we concluded that 80% of the patient in this clinical trial, they had complete response. Which is quite impressive response in terms of the fact that these patients, they had CAR therapy before, and now it didn't work for them, but now we changed this their production time from 9 days to 3 days, and also co-express IL-18, and we get such an impressive result. The other point that I would like to mention about this clinical trial is that, when we started this clinical trial, the dose that we used here was 3 million CAR-positive T cells, and compare to the FDA-approved Kymriah that we use 600 to 800 million CAR-positive T cells. So it was very minimal dose that we used for this patient, or we started the process, we started the clinical trial with those, and we got a complete response. So that gives a very strong message that necessarily number is not important. Quantity of how many CAR T cells we're infusing to a patient is not important. What is very important is the quality of CAR T cells. So we can have a low number of really high quality CAR T cells, and we can get a very impressive response compared to if you have a large number of low quality CAR T cells. So as we said, the phenotype is very important, and we know that CD3, CD28 has a strong signal that makes cells differentiate. We ask ourselves the question that can we eliminate this TCR activation? And also, we wanted to shorten the process from 3 days to even shorter, for either 2 days or 1 day. So here we thought okay, can we just expand them for 1 day? And basically that's time that we just wanted to have CAR gene transfer into quiescent T cells. So we did not want to activate these cells, and we did not want to expand cells, providing that we could have CAR gene delivery into quiescent T cells. We know that natural HIV, which is our lentivirus, is based on HIV, has the ability to infect quiescent T cells, unlike gamma retroviruses that they need cell division in order to infect the cells. HIV-derived lentiviruses, they are capable of transducing both dividing and non-dividing T cells. And there are different cytokines such as IL-7 and IL-15 that they are permissive for lentiviral mediated gene expression. So based on this data, we thought that there will be, if we can gene transfer to quiescent T cells, we will end up with more potent CAR T cells in a way that is a lot simpler and a lot more cost effective, because it would be different that we generate CAR T cells within a day or within 2 weeks. And also it's a simple process, and we can eliminate a lot of usage of some sparse material that we use, such as human serum or media, that is very important now, we know, for a generation of CAR T cells. So we tested this idea. We exposed quiescent T cells to lentivirus and followed the kinetic of transduction. And as you can see here, by 72 hours, we start to see the sign of protein expression on the surface of the T cells. And however, compared to the activated T cells, the transduction efficiency is not optimized. So for the same amount of lentivirus that we use, maybe for activated, we get around like 90% transduction, but for non-activated, we get around 10% transduction. And as expression is impaired when the cells are treated with reverse transcriptase inhibitor or integrase inhibitor, it suggests that the expression that we see is stably integrated and is not pseudotransduction, and is constitutively expressed in the T cells. We also wanted to provide more insight into the whole process, so we used flow cytometry to test the expression of our lentivirus in different subsets. And we observed that memory T cells are more readily transduced in both CD4 and CD8 subsets. But what was important for us is that we wanted to make sure that we can transduce CD4, CD8, and all T cell subsets that we have, and which we concluded that we can do that. And here we hypothesized that if we eliminate the CD3, CD28 activation step, and if we eliminate like all the expansion process, providing that if we have effective gene transfer into the T cells, we will yield with the T cells with high functional potency. So this process is published in 2022, and we compared two different products. One is our standard product, which is when we get the cells, we activate them right away, and after 1 day, we expose them to lentivirus and expand them 9 to 14 days until they finish their cell cycle. And then we wash them and freeze them or inject them, debeat them from the cells, and then inject them to mice. And the other process, we compare them with a process that we call it like 1 day CAR T cells. And this is a really, really minimal and simple process that we get the cells and then we expose them to lentivirus for less than 24 hours. And we inject this, we can inject them directly right away. So we wanted to test their efficacy in mouse models, and we use the same NAM6 mouse model of leukemia, and here we use a stress test and compared stress test, using different doses, and go to really subcurative doses and compare to the standard process that usually we use, like 3 million CAR positive T cells day 9. And this slide shows like the BLI measurement of the non-activated 1 day CAR T cells in blue compared to day 9 activated in red. So if you look at this graph, you see that these cells, the day 9 activated, they clear the tumor within a week, very quick. And however, after that, all mice relapse. And this is usually a typical response that we see now in this mouse model that we use. But when we look at the non-activated T cells, they take longer to clear the tumor. Like here, and it's very dose dependent. Here is a 2 million T cells compared to 0.7 million or 0.2 million. If we use lower dose, of course, we have less. It takes longer for them to clear the tumor. But in general, they can clear the tumor and then they can persist for such a long time, as you can see in this mouse model. Up to 90 days, we show that they can persist. And in all cases, we always check the number of T cells or CAR-positive T cells in the peripheral blood. And as you can see here, we see the number of T cells it's very dose dependent, but however, in all cases, the non-activated T cells, they have higher number of T cells and higher number of CAR-positive T cells compared to the day 9 product. So one problem that we have here is the low transduction efficiency of non-activated CAR T cells. As I showed in previous slides, that maybe for the same amount of lentivirus, we get 90% transduction for activated T cells compared to 10% transduction for non-activated T cells. So we of course thought we have to optimize this process. And in order to optimize the transduction efficiency, we need to have an understanding of the whole infection process, which it starts with interaction between lentivirus particle and cells via VSVG, which is our envelope that we use. And it's through a very well-defined binding rather than fusion events for other envelopes such as measles. So for our envelope, which is VSVG, they bind through a cell surface receptor called LDL receptor, and it's a pH-dependent process. And after they bind, the process, the next step is formation of this clathrin-coated pit, and that this promotes the endocytosis of lentivirus inside the cells, which is within a bubble, right? And then after this, the fusion has to happen so that the cells fuse in the cytoplasm of the cells. And following that, there are number of events such as uncoating, and then also reverse transcription and nuclear import, and finally integration into the genome of the cells, and following by protein expression. So for non-activated T cells, quiescent T cells, there are hurdles at each point. So it's a multi-step process, that, as I explained, it's binding, endocytosis, fusion, reverse transcription, and integration. And we have hurdles at each step for non-activated or quiescent T cells. And for example, if we compare the LDL process, LDL receptor on the surface for quiescent T cells, we have maybe around 2 to 3% LDL receptor on the surface if we compare it to activated cells that are 70 to 80% LDL receptor. And also there are hurdles in different steps. For example, for the reverse transcription process, it's very inefficient because there is not enough nucleoside inside the cells in order for proper reverse transcription process. And there are some restriction in the nuclear import and integration process. So for us, if we have this in mind, the whole process, if we want to start to optimize the process, we have to look at each step to see, how can we help this process to get, to get better transduction. So we started to optimize each step through, different mechanism to help to increase the transduction. Some of them I explained here, and there are many more ways that we up to now successfully we managed to increase the transduction efficiency. For example, one very simple way to increase the transduction, we wanted to increase the binding because, as I mentioned, there are not enough LDL receptor on the quiescent T cells. So we recognize that there's a potential benefit in a short-term serum starvation. If we serum starve the T cells, it depletes all the cholesterol inside the media, and then it results in recruiting more receptors to the surface, which in return, it helps the binding process because, we would have more receptors to the surface. And then, we show here in panel A that we can increase the transduction efficiency significantly. And also, I mentioned that there are inherent limitation in the reverse transcription process. One of the ways to overcome this, we wanted to increase the availability of deoxynucleoside. So we exogenously added deoxynucleoside into the media, and we show that we can increase the transduction efficiency again by 2 to 3 fold by just adding deoxynucleoside to the media. Another very simple adjustment that we considered was the geometry of the vessel that we transduce ourselves. So maybe the step before the binding process, in order to even binding happen, we have to have like our lentivirus particle interact with the T cells. So they have to get like in a close vicinity so that the binding process and so on, like all the steps to happen. So if you think about it, in the lab, we use these large vessels that we maybe add a drop of lentivirus to that. And for each lentivirus, they have to cover a lot of distance in order to even get close to the cell, right? Before they can bind to the cells and interact with the cells and bind to the LDL receptor and the rest of the stuff. So we just this is like a diffusion limited process. So just to help that this help the lentivirus and cells to interact, we adjust the geometry of the vessels that we're using so that they interact more with each other. And we see like by just adjusting this process, we could increase the transduction efficiency here. And when we mixed all of these different ways, and we noticed that in some cases here, from 4% we could increase the transduction efficiency to almost 50%. So there are several other ways that we also tested by now, and we know that we can increase the transduction efficiency. And we're now like doing different experiments to see if do they, is their effect is additive, so or they in parallel increase the transduction efficiency. But this process is still like under investigation. As co-expressing IL-18 gave us like a great result in our clinical trial in the 3-day manufacturing process. We thought that now we have to test that in non-activated process, quiescent process. So here, what we did, we just wanted to compare two different groups, which is cells that are, both of them are expanded only for 1 day. One is activated and one is non-activated. And we used humanized CARD19 that co-express IL-18 and compare that again in NAM6 mouse model of leukemia. And as you can see here, in both cases, we have a very impressive result in terms of, in terms of clearing the tumor. So they can clear the tumor perfectly. And sorry. But however, if you can look at the red graph, which belongs to non-activated CARD19, they have like such a quick response, and they can control the tumor. And this data is confirmed with the number of the T cells in the blood, as you can see in different days here. And here, like by day 38, we have like almost a log more number of T cells in the blood in this case. We also checked the cytokine profile of the mouse serum, and here, as you can see, in the red group belongs to activated, 1 day activated, and the blue is 1 day non-activated, and we have like higher IL-18 in the mouse serum. However, in other cases, we see, for example, for interferon gamma, even though they have a better response, we have like a lower interferon gamma in terms of like the cytokine in the serum of the non-activated CARD19 IL-18 and other cytokines. So it's very impressive the way that they can control the tumor and not generate like huge amount of cytokine. The other thing that we wanted to test the efficacy of non-activated and quiescent 1 day CAR T cells in a solid tumor. So here we used a ASPC1 PDAC xenograft model, and we used mesoCAR that are co-expressed with IL-18. And these grafts, we only, as I mentioned before, we only expose these cells less than 24 hours to lentivirus, and just very simple process. Of course, we used the optimized process, but the whole process is way simpler than standard manufacturing is available out there. That we activate T cells and expand for 2 weeks. So here is a minimal process, and as you can see again in the tumor control, we see this non-activated or 1-day CAR T cells. They can control tumor properly. They can bring it down, and it's very dose dependent. Of course, the higher the dose, the faster that they can control the tumor. But what we wanted to really see is that, can they be used in a solid tumor environment, which we showed that they have a great efficacy here. So in conclusion, I just wanted to mention that we are able to generate really potent CAR T cells that are only take 24 hours, like 1-day process, and they have and this has, of course, implication for cell therapy, because in this, we can use like a really limited dose for CAR T cells to be efficient, and the cost of the whole process is a lot lower. It's a simple process. Simple process is easy to get automated, which is very important. And also it has a very strong implication for a patient that they have rapidly progressive disease, because many times we lose patients while they're waiting for their CAR T cells to get manufactured, if they are living in an area that with the proper healthcare, that we can even offer CAR therapy. So for us, the idea and the future direction is this thinking of having like a point of care solution for CAR therapy. And the idea is that we completely eliminate this ex vivo expansion and a very complicated process, and we only have like a 1-day quiescent CAR T cells, that there is no activation. There's not, we don't need to use large manufacturing facility. We can just, in a very minimal process, we can generate CAR T cells and having like a low cost, and also not use media and other materials such as human serum, that are very sparse, and there are limited number of them out there. Limited amount of them, out there exist. At some point we get through like all the human serum that exists out there. And the other thing is, I think it's very important because it increased the accessibility of such a great therapy to more patients. If you think about it, if we need to generate CAR T cells for one patient for 2 weeks, now if we eliminate all these hurdles and have 1-day manufacturing CAR T cells, we can provide this, for more patients, which is very important right now. One of the problems that is out there is the accessibility of all patients to such a great therapy, and this is what we would like to have in the future, to be able to offer this therapy to more patients, and patients that they live in any geographical places that not necessarily have the same healthcare environment, or places that right now CAR therapy is being offered. And by that, I would like to thank everybody who is involved, and of course, like there are like way more number of people that are involved in the whole process, and we're a true believer of that the teamwork makes the dream work. So many number of people, patients, and that are involved for us to make this therapy, make this therapy to go further. And thank you.
Thank you very much, Dr. Ghassemi, for a fascinating talk. As with all the talks, we are likely going to have lots of questions from the audience, and I can see we have received some questions already. So I'll move on straight to your Q&A now. So for your first question, what are the barriers and key challenges in advancing 1-day CAR T cells to the clinics?
So as of now, there are several barriers, of course, to translate this 1-day CAR T cells to the clinic. I think one important aspect that we have to consider is defining different release assay and release criteria for our CAR T cells, which would be very different than what is standard like out there. For example, many of the testing that we do to release the CAR T product, takes 4 or 5 days, for example, the sterility test, right? And it defeat the whole purpose of having a 1-day CAR T cells if you need to just like take 5 days to just like check their sterility. Or also, there are other factors that we have to pay attention. For example, the dose of the CAR T cells, because when we have the 1 day, we do not expand T cells, so we start working with the number that we get. And however, we think these are very easily resolved, because 1-day CAR T cells, they're very potent CAR T cells. And as we saw the result of our 3-day manufacturing CAR T cells, and we started with 3 million dose of CAR positive T cells, and that's almost the same dose that we use in our mice experiment. And we got patients that they had a complete response. So these are, I think, factors that we have to start to pay attention, and not start to define new release criteria, new release assays for the CAR T cells that get generated, and not use the same criteria or assays that we use for the standard CAR T cells that is out there.
Thanks, Dr. Ghassemi. That's a great overview. So let's move on to your next question now. So as we look at the technology available today, what is the anticipated impact of current bioreactors or facility technology on the ability to generate CAR T cells rapidly? And are there any limitations with the existing infrastructure?
I really believe that in the future, we do not need the infrastructure is out there right now for, generating 2 weeks or longer product. And so this will be, to my opinion, this will be eliminated. And there are, of course, new technology has to get substituted. So, for example, many of the bioreactors that are out there, they are capable of generating really large number of CAR T cells. We're talking about billions of CAR T cells that it's not needed for this platform that we are working on, right? So in fact, we have to start to work on designing new platform that start to work that work with low number of CAR T cells, so that we can and it has to be a lot simpler, because the idea that we're thinking to have point of care manufacturing, it has to be something that is very simple and fully automated, so it's not there's no exposure of let's say human to the CAR T cells. So something that we can have a black box, that we can introduce the T cells, and then get CAR T cells within a day. We can have the patient, and then within a day they get their CAR T cells infused, and that that would of course make a huge impact. But I think those are the technologies that has to be considered. Also, different technologies in terms of having a rapid sterility test or rapid RCL test. So these are the tests that has to get developed, basically, for this platform to get translated to the clinics.
Definitely. That's interesting to hear. So now let's turn our focus to the future. I know innovation is key to advancement. So what kind of future technological advancements do you expect to see that will streamline and improve CAR T cell therapy?
Well, something that I mentioned was during the talk, that I think it's important to have high quality CAR T cells instead of the more number of CAR T cells. So the higher the higher the quality, the better the response, right? And then the higher the quality, the lower the number that we need, number of the CAR T cells that we need here. So and also, the big challenge, of course, is the solid tumor, that we have to increase the defense of CAR T cells in any aspect that we can, right, in order for them to be able to tackle the tumor. So I think in this case, what we see is that, for example, the armor CAR T cells, such as co-expressing different genes or cytokines such as IL-18, that I showed some data, that I think it will impact the quality of CAR T cells, so it can make them more potent CAR T cells. Or Also, there are designs such as, again, if you have different car design or like a very designed mixture of subsets of CAR T cells that we can infuse to the patient. For example, if you can design that we need such and such percent of effector memory cells to get like immediate response, and such and such percent of memory T cells to achieve persistence. So I think like these are in the near future, it would be more the product would be more well defined, and also I think more potent using like these armor CAR T cells approaches.
Yep, sounds like CAR T cell therapy holds great promise, but could it be even more effective in combination with other approaches? So, can you elaborate on the possibility of combining this method with an approach that remodels the tumor environment to enhance treatment outcomes?
Oh, yeah, of course. For example, we can all the effort that we do here is just to design and change our CAR T cells to better and more potent, right? But at the same time, what we can do, we can remodel the tumor microenvironment so that CAR T cells can penetrate, and they can, it's like again, like it's a teamwork. We to a point, I think like we can make our CAR T cells better and better, I think, but it would be stronger effect if we can also remodel our tumor microenvironment so that in combination they would have a better efficacy.
Thank you so much, Dr. Ghassemi. This definitely opens up exciting possibilities for the future of cancer treatment. However, that is all the time that we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Ghassemi, and we'll be back soon with the next talk.
Thanks.